Hello students, today we are going to discuss about DNA metabolism. Now, what are the key topics we are going to see in detail? That includes DNA replication, DNA repair, and DNA recombination. First of all, we'd like to know what is meant by DNA metabolism. For a copy of DNA is synthesized with high fidelity before each cell divides. The error that is arise during as well as after synthesis of DNA are constantly checked and repairs are made, and the segments of DNA are rearranged either within the chromosome or between two DNA molecules, giving offsprings a novel DNA in this case. The DNA metabolism consisting of various set of enzymes catalyzed and tightly regulated process that is achieved by the various tasks. Why we are to synthesize the DNA in an intact form? The reason is that it is passed on to the next generation from the parent to the younger ones. So it has to be synthesized carefully, as well as the error which are arising during the replication mechanism has to be constantly checked and the repairs to be made. And in order to bring the new combination of phenotypes, the DNA containing the genes has to be rearranged within the chromosome, and the segments of both parental genes are exchanged for that sake to provide a novel form of DNA to the offspring. What is DNA metabolism? The DNA metabolism consisting of various topics: DNA replication, DNA repair, and DNA recombination. DNA replication, in which the copies of DNA molecules are faithfully made. That is, copy of parental DNA is made. That is, as such, the parental copy are not passed to the next generation. Copies of chromosomes are made, then it is passed to the next generation. That too, it is in the form of semi-conservative mechanism. That is, one parental DNA copy, another daughter DNA copy is passed to the next generation, another set is retained within the parent itself. In DNA repair, if any damage happens in the case of the DNA, it has to be attended, and the integrity of the DNA has to be maintained by the process of repair mechanism. And DNA recombination, by which we get DNA sequences or rearranged within the chromosome to provide novel DNA combination in the offspring. This is a map of the E. coli, that is prokaryotic chromosome, for your kind reference. We know very well the DNA replication is semi-conservative. That is, one parental DNA, one offspring DNA is passed to the next generation. It is proved with the help of isotopes, that is nitrogen 14 as well as 15. Nitrogen 15 is after that it is centrifuged DNA polymerase, the enzyme, in the presence of which we require primer, parental DNA template, all four deoxyribonucleotides, that is DNTP, that is normally the DNA contains four nucleic acids, that is adenine, cytosine, guanine and thymine. And a divalent cation such as Mg2 plus ion is necessary for the process of our DNA. Here the diagram shows the template DNA, that is parental DNA, and the incoming nucleotides are added according to that. For example, if the parental DNA contains thymine, a complementary of thymine, that is adenine, is added to the growing strand of the daughter, that is especially to the primer it is added. Then if it is cytosine, means the guanine will be added, or vice versa. So normally then incoming nucleotide is added to the 3-hydroxyl group of the existing primer for the formation of the phosphodiester linkage. Here is the mechanism. The parental DNA serves as the template, and the nucleotide is triphosphate, serves as the substrate for the synthesis. The hydroxyl group of the growing chain, that is especially in the case of the primer, makes the bond for the alpha nucleus, alpha phosphorus of the nucleotide, and pyrophosphate is leaving during this procedure. Here we can see the diagram how the template strand containing incoming nucleotide creates a phosphodiester linkage with the existing 3-hydroxyl group for the formation of the phosphodiester linkage. How the DNA polymerase containing active site aspartate amino acids and Mg2 plus ion, that is a covalent cation that is responsible for the formation of the phosphodiester linkage, and the pyrophosphate is eliminated, is shown in the diagram. Normally synthesis of DNA does not occur continuously on the both the strands of the parental DNA, even though the parental DNA is 5' to 3' as well as 3' to 5', that is parallel and anti-parallel in nature. Once the daughter DNA is synthesized, the direction of replication always happens in the case of 5' to 3' direction only. In the leading strand, that is in the 5' to 3', it moves continuously because the direction of replication is 5' to 3', whereas in the lagging strand, that is 3' to 5' parental DNA, the DNA is copied in fragments, that is in the reverse direction, that is in the 5' to 3'. These fragments of DNA are called as Okazaki fragments. The base selection by the DNA polymerase is fairly accurate. About 1 error per 10 to the power 4 nucleotide happens. Unfortunately, we have the proofreading mechanism in the case of the DNA polymerase with 3' to 5' exonuclease function, which is having the highest accuracy of 10 to the power 2 to 10 to the power 3 fold, that can able to proofread and remove the mismatched nucleotides or mistaken nucleotides. Mismatch repair system, always the mismatched base or the nucleotide or mistaken nucleotides are removed after replication, and further that improves the accuracy of the DNA. Why this is done? The reason for removal of the mismatched nucleotides or wrong nucleotides occurs in the case of the DNA to maintain the integrity of the DNA, because the accurate DNA has to be transferred from the parent to the next generation. So this is the structure of the DNA polymerase 1. When the DNA polymerase 1 is made up of 928 amino acids. When it is digested with trypsin or substilin, it is fragmented in two domains, the larger as well as smaller fragments. The larger fragment is called as clinofragment and the smaller one is consisting of 5' to 3' exonuclease function. The molecular weight of the larger fragment is about 76,000 daltons, whereas smaller fragment has molecular weight of 34,000 daltons. So in the larger clinofragment, it has polymerase function, that is 5' to 3' polymerase function, where it combines two nucleotides by its polymerizing activity. Whereas in the case of the another larger clinofragment, another function is 3' to 5' proof reading function is also available. Whereas in the smaller fragment, it is consisting of 5' to 3' Nick translation function. Here is the 3' to 5' exonuclease function of the DNA polymerase 1. The DNA polymerase is far from relevant. It is responsible for cleanup function during the replication process, recombination as well as repair. The DNA polymerase 5' to 3' 1 containing 5' to 3' exonuclease function is distinct from 3' to 5' proofreading function because it creates a cut in the DNA nearer to the primer. We call it as nick. Then later the RNA is removed. Then further the nucleotides are added by the DNA polymerase 1 by its polymerase function. The gap which is created is after coming closure, it is sealed by DNA ligase. This is the structure of the larger clinofragment of DNA polymerase 1 that is having polymerization and proofreading function. The 5' to 3' exonuclease activity. The clinofragment is found to contain the 5' to 3' exonuclease activity that permits to extend the DNA strand even if the template strand is already paired. It is responsible for degradation of the DNA or a segment of RNA which is paired to the parental DNA. But DNA polymerase 3 lacking this 5' to 3' exonuclease activity. The second enzyme that is associated with replication is DNA polymerase 2. The DNA polymerase 2 is a minor component of cell during the normal growth, but it is inducible by SOS response. That is, when there is a damage to DNA by the action of ultraviolet radiation or by mutagens or any chemicals or radioactivity like alpha, beta, gamma radiation. On such conditions, when the damage is happening in that region, it loses the base and creates epicytes, that is apurinic or apyrimidinic sites. That is removal of purine as well as pyrimidine bases called as epicytes. The DNA polymerase appears at that time for the DNA repair function. This enzyme is responsible for base excision repair and nucleotide excision repair. What is my base excision repair? In which every cell has a set of enzymes called the glycosylases that is capable of identifying the damage in the DNA lesion region and especially it removes the glycosidic bond by breaking it, which connects the base purine or pyrimidine base with that of the deoxyribose which is present in the DNA. Once the base is removed, when the purine or pyrimidine base is removed, that region is called as apurinic or apyrimidic site in the DNA. It is also known as AP site. Then further, the DNA polymerase action comes into play, that is responsible for filling the gap using the supplementary strand which is present in it. Whereas in the nucleotide excision repair mechanism, the lesions are identified. They are looking like aspartate breakers, that is removed with the help of a specific type of enzymes called as ABC nuclease. This ABC nuclease is made up of 3 subunits which is coded by the genes UVRA, UVRB and UVRC genes. This enzyme is capable of identifying many types of lesions, including the cyclobutane pyrimidine dimers and 6,4 photoproducts which are created on exposure to ultraviolet radiation, and base adducts which are created. These base adducts are in turn excised with the help of this ABC nuclease. It is totally different from the action of the endonuclease and helps in maintaining the integrity of the DNA. These enzymes are referred as SOS response enzymes. The reason is that they act like ambulance at the site of accident. Same manner, these enzymes appear at the site of damage. Here is a comparison of 3 DNA polymerases of E. coli. This polymerase 1, 2 and 3. Now we are going to see the largest polymerase, that is DNA polymerase 3, that is coded by the gene Pol-C or DNA E. It is made up of more than 10 polypeptides and the molecular weight is the DNA polymerase 3 has several subunits which includes beta, gamma, delta, delta dash, epsilon, psi and tau subunits. Here is the composition of the DNA polymerase 3: alpha, epsilon, beta, tau, gamma, delta, delta dash, psi, pi and beta. It is consisting of 2 regions, core enzyme as well as apoenzyme. Together we call it as holoenzyme. The 2 cores are consisting of a clamp loading complex called as gamma, which is made up of tau 2, gamma, delta, delta dash and additional protein psi and pi, and holoenzyme is coenzyme with beta subunit. Together we call it as coenzyme. The gamma is also known as clamp loading complex, which is responsible for loading the beta subunits on the lagging strand as well as leading strand of the every Okazaki fragment. Here is the structure of the DNA polymerase 3. Here the definition for coenzyme and holoenzyme. The whole definition for coenzyme is only those subunits that are required for Basic enzymatic activity is called as core enzyme, whereas holoenzyme consisting of the units that are required for enzyme activity as well as regulation. The core enzyme consisting of alpha, epsilon and theta. Alpha is responsible for the 5' to 3' DNA polymerase activity, whereas epsilon is having exonuclease proofreading activity, whereas in the case of the core enzyme, the beta is a sliding clamp, whereas the clamp loader complex is alpha, delta, delta dash, psi and pi. The processivity switch is found in the case of the tau complex. Here we can see the structure of the heterozymer component of DNA polymerase 3. DNA B is the helicase that is responsible for unwinding the two strands of the double helical DNA, because normally the DNA is twined over the other because of the base pairing nature in the complementary strand containing nucleotides. It has to be separated for the process of copying. The core is made up of alpha, epsilon and omega, and here we can see the beta clamp that is loaded with the help of tau, gamma, delta, delta dash. In the figure we can come across the open and closed form of the beta clamp that is responsible for binding with the leading as well as lagging strand, that is both the strands of the DNA. The two subunits of polymerase 3 from a circular clamp that surrounds the DNA. This diagram clearly shows how it is attached with the tougher double helical DNA. Further, the DNA replication requires many enzymes and protein factors for the process of replication. That includes helicase, that is responsible for removal of or separation of the DNA duplex. Topoisomerase is responsible for relieving the twist which is present in the case of the DNA. There are two types of topoisomerase are topoisomerase 1 as well as topoisomerase 2. Topoisomerase 1 is called as DNA gyrase, and further we come across various single-stranded binding protein. Once the DNA is separated, it cannot stay back because of its complementarity nature. Again, the hydrogen bonds is formed between both the parental DNA and it goes back to the original structure. To avoid that, the single-stranded binding proteins are responsible for stabilization process once the DNA is separated by the action of helicase. In order to start the replication, we require a primer. As such, the DNA polymerase cannot add the nucleotide by its polymerase activity. It needs an anchor or support to which only it can add the nucleotide, in which the RNA is added as a primer. In the leading strand, there is only one primer is present, whereas in the lagging strand, since it is synthesized in the discontinuous manner in the form of fragments, we call it as Okazaki fragments. Every Okazaki fragment contains RNA that is in turn synthesized by the enzyme called as primase. Because it only responsible for the initiation of the replication, it is called as primase. Further, the DNA polymerase 1 by its nick translation process removes the RNA and fills the gap between the adjacent Okazaki fragments by its polymerase function. The adjacent Okazaki fragments cannot be sealed by itself, so we need a sealing enzyme called as ligase, which is capable of sealing the adjacent Okazaki fragments during the process of replication. These are all the various enzymes and proteins which are necessary for the replication process. So replication process we are going to see in detail. The replication of E. coli chromosome is an enzymatic process in which the daughter DNA is synthesized from the parental DNA, which is identical to the parental in nature. The rate of replication in the case of the prokaryotic cell is 1500 nucleotides per second. Imagine, to complete the replication of entire E. coli genome, it takes only 40 minutes. In the system, we have 23 pairs plus XX or XY. XX in the case of the female and XY in the case of the male. So we have this much amount of chromosome as a total nucleotides, about 3.6 into the 10 to the power 9 billion base pairs are formed. So how long it takes to complete a replication in the case of eukaryotic system is the rate of the replication in eukaryote is 10 to 100 nucleotides per second. It's slower when compared to the prokaryotic system, and moreover it takes the complete replication process to be over by 6 to 8 hours. In prokaryotic DNA, especially in the case of the circular DNA, only one replication fork is present, whereas in the case of the eukaryotic system, several points where the replication fork begins, that is separation of the parental DNA takes place, and where the enzymes and proteins are recruited for the process of replication to be over. Just like constructing a bridge at several places to complete the process at the quicker time. Same manner in the case of the eukaryotic system, at several points the replication will begin and it will be concluded. The space between the two replication forks in the eukaryotic system is about 20 kilobase pairs apart. The process of prokaryotic system is broken into three major steps: initiation, or E site. This is made up of 245 base pairs. The origin site is consisting of 245 base pairs, which signals the DNA polymerase to come and join and to start the process of replication. Replication origin. This oricite is made up of 13 base pair repeats as well as 9 base pair repeats are found. So nearly 3 such 13 base pair repeats and 5 such 9 base pair repeats are found in the case of the origin site. That is a highly conserved in nature, and it is a consensus sequence. That is a repeated sequence, which is these consensus sequences will have such repeats. That is, in the case of 13 base pairs, it is GA, GA, sorry, GATC, TNTT, and TTTT. Whereas in the case of the 9 base pair repeats, TT, AT, TN, CA, CC. This replication beginning point or the origin point is also called as a replicon. It's a unit of genome in which the DNA is commencing the replication process, to which various enzymes and proteins are recruited to form the pre-primer complex to initiate the replication process. Normally, the origin site is situated 74 minutes nearer to the ILV gene, and this origin site consisting of 245 base pairs, already I told you, that is 13 base pair repeats and 9 base pair repeats. These are highly conserved sequences. Conserved means which cannot be seen elsewhere. That is why we call it as highly conserved sequence. It's common for many of the bacteria. So various proteins that are recruited at the site of origin for the process of initiation of replication or starting up of replication process, that includes DNA A protein that recognizes this origin site containing 13 base pair repeats as well as 9 base pair repeats. Further, the DNA B protein, that is helicase, comes and joins for the process of unwinding double helical DNA into separate DNA, individual DNA, parental DNA template. And this is assisted by DNA C protein. The DNA B protein is assisted by DNA C protein. Further, histone-like protein, HU-like proteins are also associated. In addition, FIS protein, IHF protein also responsible for stimulating the initiation process. Then, once the initiation complex is formed, the primase comes and joins that starts synthesizing RNA primer in the 5' to 3' direction, to which only the DNA polymerase can add the nucleotide for the process of replication, irrespective whether it is in the leading strand or in the lagging strand. The single-stranded binding protein is responsible for maintaining the parental DNA in a separated condition by it is attaching on the surface of the separated DNA. And DNA topoisomerase is responsible for removing the torsional or twist which are present in the case of the DNA which moves in the forward direction prior to our DNA helicase. Let us see how these proteins are recruited for the process of initiation. The DNA A protein recognizes and binds to 4 9 base pair repeats in origin site to form a complex of negatively supercoiled origin site DNA. Here which the central core of DNA will be present as monomer and is wrapped by the DNA, and further this is recurring in the presence of histone-like protein that facilitates bending of the DNA. Once the DNA subunit successfully melts the 13 base pair repeats, it forms a open complex. Approximately 22 base pairs are present at that region. Since this is rich with AT, it is easy to break the 2 hydrogen bonds in that region. Instead, GC. Once the DNA protein binds, it further guides our DNA B, that is DNA helicase, that is loaded by the DNA C. The DNA C is called as helicase loader, that is recruited at the melted region to form the pre-prime complex. Here we can see from the diagram, for this pre-primer complex formation, it requires the energy that is in turn provided by binding with ATP. Once the DNA B is loaded by the helicase loader DNA C, the ATP is hydrolyzed and it gets converted to ADP and PI to form the open complex or pre-priming complex. Next step is elongation. The elongation involves various proteins that includes single-stranded binding protein, DNA helicase, primase, DNA polymerase 3, 1 and ligase and gyrase. The single-stranded binding protein is SSB, binds to the separated DNA, whereas helicase unwinds and forms a pre-primosome complex. Primase, that is responsible for RNA primer synthesis in the leading as well as lagging strand, that is Okazaki fragments. The elongation of the DNA is done with the help of DNA polymerase 3. It adds a nucleotide to the primer and further the filling up of gap and the excision of primer in the leading as well as each Okazaki fragment of lagging strand is done by DNA polymerase 1. The gaps are sealed by DNA ligase. Here is the diagram which shows the elongation process of synthesis. The DNA polymerase, DNA topoisomerase, that is DNA gyrase, that moves in the forward direction, and we can see that the leading strand is synthesized continuously along with the replication fork movement, that is 5' to 3' direction, whereas in the lagging strand it is a discontinuous synthesis because it runs in the opposite direction, and we can see the attachment of single-stranded binding protein. Just it is attached on the top as well as at the bottom. As the enzyme migrates, whether helicase or primase, when it migrates, it falls off and again it takes up new position of unwounded region of the DNA, that is by the single-stranded binding protein. The model of DNA on the leading and lagging strand by the DNA polymerase 3. Here we can see that the clamp, beta clamp of the DNA polymerase 3 is bound with the leading as well as the lagging strand. Whereas in the case of the lagging strand, you can see the RNA primer is synthesized for every Okazaki fragment with the help of primase. And the clamp loaded complex opens the beta sliding clamp and further closes for this process. The synthesis of leading strand is continuous once the DNA is unwound by DNA B helicase. The leading strand only one primer is synthesized and continuously the nucleotides are added by the DNA polymerase 3, whereas in lagging strand, since it runs in the opposite direction, the unwinding it takes. Initially the length of the DNA will be parental DNA will be very smaller. As the helicase moves faster, further nearer to the replication fork, the DNA loops out and new primer is synthesized and further it is extended by the DNA polymerase. So the Okazaki fragments are synthesized nearer to the fork region only. Once the Okazaki fragment is synthesized, the unbounded region, again the DNA bounded region is looped out, the primase binds and a new primer is synthesized and further it is extended by the DNA polymerase 3. During this time, a new beta clamp is loaded in the newly synthesized primer with the help of DNA polymerase 3. So the synthesis of new Okazaki fragment is completed. The process repeats for every single fragment of Okazaki. A new primer is synthesized and a new beta clamp is loaded. And the DNA is looped out to fix its position because it has to complete the processivity of replication in both leading as well as lagging strand in the same length of the DNA. Once the new beta clamp is loaded onto the new template for the primer by the clamp loader, the synthesis of new Okazaki fragment gets completed on the lagging strand. The lagging strand core subunits are then transferred to the new template primer and its beta clamp. The old clamp is discarded. Once the DNA replication is over, what happens? The DNA, that is DNA polymerase 1, removes the RNA primer of every Okazaki fragment and it fills the gap. This removal of primer is done with the help of our NIC translation, that is the 5'3' exonuclease function. It removes it and the gap is filled by the 5'3' polymerase function of the DNA polymerase 1. Once the adjacent Okazaki fragment primer gap is filled, then comes the role of DNA ligase. This is the sealing enzyme. DNA ligase is adenylated DNA ligase. That is AMP is added from the donor ATP, and this adenylated DNA ligase then acts upon the adjacent Okazaki fragments containing region of gap. It carries out transfer of a phosphate group to the nicked region containing DNA. Once the phosphate group is added, it carries out a nucleophilic attack on the hydroxyl group of the previous fragment and gets sealed. And during this process, AMP is released after sealing is over. And once all this is over, the replication comes to an halt when opposite forks comes and meets in the case of the circular DNA, because it is a bidirectional replication. When counterclockwise and clockwise forks comes and meets, automatically the replication comes to an halt in the case of the E. coli. Normally, the termination of the replication requires a special type of protein called as TUS proteins, or we call it as TUS, termination utilization substance protein. There are various types of TUS proteins are available. There are A, B, C, D, F and G. There is no E. So counterclockwise traps are A, D and B, whereas clockwise traps are B, C, G and F. By sensing these TUS proteins, the replication comes to an halt. Whereas in the case of the linear DNA, they are totally different. The replication is. The terminal point is consisting of separate sequence called as terminal site, where the DNA polymerase recognizes and it comes to an halt. This is the story of prokaryotic replication. We'll see the eukaryotic replication in next video.